Hey, today's session is going to be over forensic genetic genealogy, and our presenter is Dr. Wilson. He is a retired supervisory special agent with the FBI and is now an associate professor in the forensic science program at George Mason University. His area of expertise is in forensic science DNA, forensic DNA analysis, including DNA sequencing, mitochondrial DNA, probabilistic genotyping, and interpretation. I now present to you Dr. Mark Wilson.
Okay, thank you, Jasmine. Appreciate that. Thank you for the invitation and the opportunity to share some pretty exciting information with you on an emerging technology called investigative genetic genealogy. So what I want to do is give you an overview of kind of how it works, talk about some existing DOJ guidelines that seek to expand and also limit the application of the technology. And then I'll switch and talk about SNP databases for law enforcement agencies to use. Talk a little bit about laboratory accreditation and quality assurance standards that are in place and emerging in this field. And then on the technical side, I want to talk a little bit about DNA extraction and how to make a vibrant and robust library for sequencing. This is technologies that have come a long way in a short period of time and have definite implications and improvements on how this technology is used. I'll touch briefly on some existing technologies, especially microarrays versus DNA sequencing technologies and the importance of improvements there. And then lastly, I'll talk about funding sources, federal, state, and local funding sources for obtaining funds to do this. So this is a brief output of a data file. And so what you see here is an ID on the left, the chromosome number next, and then a position in what's called a genome build. That's just a number that associates back to a position in the genome. And then on the far right, you see the genotype. And that's just two letters, the four bases, it's either AA, GG, etc. So importantly, there's a tremendous amount of information that accumulates from these analyses, as opposed to the existing technologies, which typically return about 20 markers, sometimes more, 27 to 30 markers. But these SNPs, they go on and on for pages and pages. So an output for a genotype of a person, and this is a microarray output, would be about 850,000 rows of data. So it'd be thousands of pages. So there's a lot of information here, tremendous amount of information, and it's orders of magnitude greater than the information that we currently use with DNA typing, because we're looking at variation across the entire genome. So if you look at the human genome by chromosome, you can see the well-known markers, and then you see the X chromosome kind of in the center. So these are arranged by size. And then they go down and you have the Y chromosome there at the far right. So a lot of human genome sequencing has taken place in the last 30, 35 years. And the data that has accumulated has been cross-compared across different individuals to identify SNP variants, single nucleotide polymorphisms, like we saw in the last slide. These are the types of variants that can now be assayed, and it's across the entire genome. And that's just a lot of information. So the genome itself is about 3.3 billion base pairs, and that's a lot. And the SNPs occur on average about 1 in every 1,000 base pairs. So that means there's about 3.5 million SNPs in the human genome. There's a lot of real estate here and a lot of potential variation that can be used for identification purposes. The trick is to capture all this information from a forensic sample, and it's difficult. It's not easy. Forensic samples are often degraded. They're limited in their quantity, but the biggest problem is the degradation. If the DNA is degraded, it's really hard to get out the information that you expect from a pristine sample. But the importance is that because these technologies can generate so much data on a large scale and relatively inexpensively, the cost is coming down all the time. It's now possible to genotype someone and get really over a million of these SNPs in one run. And so the technology has really driven the application of this science. So when we look at the total amount of shared DNA, this is a typical output showing segments that are shared in orange. And the metric here is something called a centimorgan. And this is about 300 centimorgans shared. And that's typical for a second cousin relationship. So if you look at second cousins and you map the SNPs that are identical across the whole genome, what you see is there are segments that are highlighted in orange where there are definite overlaps in size. So there's a number of different blocks. You see that some chromosomes, like from 6 to 12, they have no shared segments. And then chromosome 17, just by chance in this particular comparison, 17 has a lot of overlap, most of the chromosome. So it's really by chance, and it has to do with, meiotic recombination and how chromosomes line up and they swap segments during the formation of sperm and egg. And so what that means is that people that are closely related, you'll be able to see these shared segments. They'll just highlight for you as shared segments. And the more shared DNA, the closer the kinship relationship. As you get further and further away, the size and the number of these orange blocks will go down to almost nothing until finally you get to something like this. Well, you can just imagine that there is a graph with absolutely no orange, all blue. And that would be no detectable relationship because there's no overlap. But here, there's quite a bit. And again, this is second cousin, so there's quite a bit of shared DNA in terms of segments. So the first case, the famous Golden State Killer case, is almost 6 years old now, April of 2018. And this was solved by uploading profiles to public databases and looking for relatives. And once those relatives were identified, then genetic genealogists could ascertain which families were involved. And by looking at combination of genetic records and traditional genealogy records, they could narrow it down, usually to a family. And from that family, then investigatively, they could go further and identify, in this case, the assailant, Joseph DeAngelo. So this really got the process started. And of course, a lot of excitement because it was a new way of using DNA. Rather than having a profile in the lab that you compare to a suspect, now you can just take a profile itself and there's no suspect involved. It's a cold case, for example. You've tested a lot of individuals, but no one's matched. So now you use these databases to upload the profile, look for family matches, and then build the family trees. And from those family trees, then work your way down to the number of possibilities. And then investigatively, you can look at these outcomes and kind of focus your investigative attention on certain individuals. So let's switch over now and talk a little bit about the DOJ guidelines that came out in September of 2019. Here's the original publication. These are interim policy guidelines, and it gives you kind of an idea of how the DOJ is thinking about this emerging technology. So one important concept is that they feel, and almost all the folks in this field, the practitioners feel that this is background information. It's almost like research, and it's there to generate investigative leads for investigators. So it's not considered to be the same level of laboratory analysis that, for example, STR typing or mitochondrial sequencing is. So it's not subject to the same types of regulations because it's lead generation, and eventually the idea is to lead the investigators to a person or persons that can be typed using traditional markers. And then those traditional markers can be compared directly to the crime scene DNA profile. And that's important because the backstop to all this family research is you hope to get a individual that you can reach out to or collect DNA from surreptitiously, and then have that typed, and that will lead to either a partial match or a full match. And then that can be used to support a warrant for an actual law enforcement collected sample using all those safeguards that are in place. So it's lead generation, it's background, it's almost, you can consider it almost as research. And a lot of people call it research when they're reaching out to discuss their findings. They will say things like, well, our research has determined such and such. But the other way that it's characterized is to generate investigative leads for only a certain number of crimes. Not all crimes. Typically now property crimes are not included. So it's rape, violent sexual assault, homicide. So here's another statement. Excuse me. If a suspect is identified after a genetic association has occurred, then the traditional typing markers come in, the STR DNA typing. They must be performed and then directly compared. And they emphasize here that if there's an STR profile available, that they expect that it be uploaded to CODIS and search for a match. And there's a lot of CODIS uploads that do not result in matches. So these are considered, some of these cases are considered dead ends or cold cases, but the DOJ expects that the profiles are uploaded to CODIS to make sure that there's no match there before embarking on genealogy. Also importantly, part of this testing is testing of what are called third parties or kinship testers. You'll see it used a number of ways, but this is usually conducted towards the end of an investigation when the genealogists have narrowed down the possibilities, but there's still some ambiguity in their results. And what they're saying is, if we could target test a certain individual or individuals and get their profiles, upload their profiles into the databases, we could then see how they relate to the unknown sample. And if they relate in a very close way, that's good. If they're further away, that's still information that helps the genealogist focus in on the areas of the family tree that they feel are the most probative areas. And so it can be used to completely, prune off big parts of the tree, or in some instances, it can get very, very close. But either way, it's information that the genealogist can use to refine the relationships, to refine the exact relationships of the people that are in the databases, because they will be cross-compared to the kinship tester as well as to the unknown profile. So you have a distance to the unknown profile, but you also have distances to other folks in that family tree that can be further refined, and those relationships are often very helpful. So when you see a case get to the kinship testing phase, what it usually means is the investigators are pretty close. The genealogists and the investigators are getting close, and then and that can bear fruit very quickly. So this is a target kinship test. So on the right, you see this is an Illumina bead chip. It's a microarray. It's microarray technology. This is the technology that is used, for example, when you spit into a tube and you send it into Ancestry or 23andMe or MyHeritage. They will load that DNA sample after they've extracted it from your saliva. They will load that DNA sample onto the chip, and these chips then generate the 850,000 SNP markers, which can then be uploaded into a database to further refine the relationships. So as you can see here, the kinship tester is not a match on the left or is a match on the right. And either way, that gives the investigative genetic genealogist more information to go on and more resolution of the tree. So targeted kinship testing is very important. And it usually means that the investigators are getting pretty close. So here's an example of a kinship testing outcome. You see here an analysis was done, a kinship analysis was done between an unknown and another sample. And a second degree relationship was obtained with very high probability. Almost all the probability falls on that second degree relationship. And this is very valuable information because, again, it refines those family relationships within this wider context and narrows down the possibilities. Okay, so let's talk about the SNP databases, the actual databases that are used for law enforcement searching and law enforcement agencies. So there are a number of them. And one of the main ones that gets a lot of attention is the GEDmatch database. And the GEDmatch database, they will partner with law enforcement, but they also have a number of different tools that can be used, bioinformatic tools that can be used for the genealogists doing the searching. And these tools are developed for folks to use on their own. The philosophy of the GEDmatch database is to give people tools to do their own ancestry research. So that's a very important one. But there's others, many others, and the two that are approved for law enforcement uploads are only two. There's GEDmatch and Family Tree DNA. GEDmatch has over 1.5 million profiles. With that database, you have to deliberately opt in. And these numbers are a little bit dated. It's more like closer to 400,000 now that are opted in. But as you see on the side of the slide that under Family Tree, there's more profiles in the Family Tree DNA database. And importantly, you're opted in to law enforcement searching automatically. You have to tell the Family Tree DNA administrators that you do not want to be used for that purpose. If you don't say anything, then you're opted in by default. So that's a very powerful database that's used in this research. So there's really two, GEDmatch and Family Tree. It turns out that with the GEDmatch terms of service, if you're looking at identifying an unidentified human remains case, say from a bone sample that was recovered, those are not subject to the same restrictions as the criminal uploads. So with a UHR case, you can use the entire database in both GEDmatch and Family Tree. Okay, this is an important slide because it highlights something, which is actually two things. One is when these databases are searched, this is the output that comes back, something that looks like this. And what you can see is on the bottom, you see some numbers like details and then total centimorgans. That's the number of those overlapping segments. So you have a pretty close match here at 980. And then the largest segment is 59.5, etc. But what you don't see in these match lists is there's no SNP data. Your actual genetic information is protected. The only thing that's released is your matches and how close you are related to each of those matches. And then also it's blocked here so you can't see it. The email address or the email alias of the donor is provided. And this is the information that the genealogists use to begin to conduct their research. So a couple of important things is, in terms of genetic privacy, none of your actual genetic data is released when these investigations are done. The investigators and the genealogists, they cannot tell what your SNP state is at any marker. They don't know anything about that. They don't know, which chromosome contains the most matches. They don't, by genotype, they can't refine it down to genotype and look at the actual data. What they can see, though, is how many shared segments and how large are those shared segments. And from there, they can get a lot of useful information. You can see also that in some cases, X DNA, DNA matches on the X chromosome are important. And X chromosomes segregate differently than Y chromosomes through families. And so that's also important information to the genealogist to tell on which side of the family, either the paternal side or the maternal side, is this match likely to be. Okay. Some other information is there, like the mitochondrial haplogroup up top, the Y haplogroup. That can be helpful as well. So any kind of information, genetic information, can be helpful. If there's a match, a mitochondrial DNA match, that means there's a maternal inheritance component. If there's a Y chromosome haplogroup match, that means just the opposite. It's a paternal group. So if that information is available, it's also displayed. GEDmatch has some very important and powerful tools. This is just one of them called clustering. And what this is showing is all the matches to a particular unknown and how they match each other. So these are internal matches in the database, matching to each other and also matching to the unknown. So the trick here is the genealogist will take this information, use these tools to try to clump these squares, these boxes, together into larger and larger coalitions or condensations of these boxes. So what they want to do is they want to reduce the number of boxes, the total number of boxes, so that they can see more distant relationships and then how these groups are ultimately linked together. You see that some are very small. That purple one in the center is very small, only a few individuals, but there is some connection to the others because they're showing up on this clustering diagram. So this is an example of one of the tools out of many that are available to genealogists that are trained in how to use these tools in GEDmatch. Okay, so here's a typical family tree. And what you can see is this is just made up people. These are not real individuals. This is a made up pedigree. But you can see the pedigree has some matches that are highlighted in red boxes. And then underneath those matches is the amount of shared DNA. So on the far left, you see Donald Larson, 54.9 centimorgans. And then there's another one underneath that, Linda Zimmerman, 34.7. These are distant relationships. And then you can see that some of them don't share any DNA with each other. So that's interesting. That factors into the genealogist's tree building and their thinking. And then you can see that in this case, on the far right, the genealogist has recommended a potential kinship tester out here, Kathleen Martinelli. And what they've found is that if this person would agree to be tested and upload their profile into Family Tree or GEDmatch or both, then the matches can be ascertained and the exact relationship between the suspects, the kind of the bubbly yellow cloud-like thing is potential male suspects. Those relationships can be further refined once that potential kinship tester has tested. And so they carefully go over everything that they can discern about an individual and try to figure out maybe who might be agreeable to do this type of analysis. And then they will approach them, law enforcement will approach them, explain exactly what they're trying to do and get their approval, because one of the strengths of this technology is everything is voluntary. All the matches in the database have agreed to law enforcement testing, either by default or obviously through saying yes. So there's nothing is compelled and there's no force here. It's all just done in research mode, but also open. Everything is open and above board, and there's no compulsion. No one is forced to give a sample into this database, and it's a good thing because it provides a lot of confidence to the people that are participating in these databases. They want to help, and they often do help, and they fully understand any risks when they're approached and asked to give a sample. And if they agree, fine, they sign a waiver. Sometimes they don't. They don't agree, and that's fine too. Yeah, so this is kind of the way that a potential kinship tester can be identified and then tested, and then all these red boxes can be expanded that you see on the left. Those red boxes would be expanded onto the right side of the tree. And you generally see in this tree, there's a paternal side and a maternal side. So a lot of the skill comes into determining which side, which matches are maternal and which matches are paternal. And sometimes there's endogamy, there's shared DNA between the two lineages, um, which also has to be factored in to any kind of calculation about the distance between any one of these boxes and the unknown. Okay, let's talk briefly now about laboratory accreditation. So microarray testing is generally done in a completely different environment than traditional forensic testing. Forensic testing is shown on the bottom, and that is regulated by ISO, and in the US, the ANAB. And the general qualifications and accreditation requirements for forensic labs are what's called 17025. That's the basis of how forensic laboratories are accredited, how they're audited, the standards that they have to comply to, how they're reviewed, etc. Proficiency testing, training, continuing education, all that is part of the forensic world. In the clinical world, they have their own sets of standards. They're just different. And so you'll see things like a Clinical Laboratory Improvement Amendments, CLIA, or College of American Pathologists, CAP. Those are the accrediting bodies in the clinical world. So because we're using microarray technology, we are falling under the clinical umbrella of quality assurance and accreditation. And that's a good thing because, even though they're different, they are held to standards which include proficiency testing and training. And so we can have some kind of assurance that the testing is being done in a high quality manner. But they are different. So marrying the two together, it kind of remains to be seen how in the long run, these two essentially different types of accrediting organizations will come together to monitor and assess the entire process, um, whether it's a forensic case or not. So here is just a list of all the quality assurance standards for DNA typing laboratories as put forth by the FBI laboratory. So there's a lot of different things: the program, organization and management, personnel, etc. And many of these will apply to the clinical world, but not all of them. And so another question is, how many of these things are important, since this is considered to be background research and is backed up by an STR match at the end of the process? What applies and what can be maybe modified? So for example, proficiency testing, obviously, that's very important. Personnel and training, those are also very important, because you want to have people that are proficient, they know what they're doing, and that just leads to very high quality results. So in the long run, the existing standards and how those are going to be married into the emerging standards is something to fully be worked out. So for example, how are the lab analysts going to be tested? Are they going to fall under 17025 or potentially CAP CLIA? What about the bioinformaticists, the folks that look over the data and make the kinship associations, or upload to the databases and look at the matches? Folks that write code for dealing with degraded DNA, etc. How are they going to be tested? It's still an open question. As is, what about the genetic genealogists? How are they going to be tested? Are they going to be assessed by other experienced genealogists, or they're going to be subjected to proficiency testing? And how's that going to work? Turns out it's very difficult to set up a blind test or a genealogical test when you're not using real people's names. And a lot of these tests, some of these genealogists know about the outcome, so it's not really a good test. So that remains to be an ongoing work in progress. Typically now the senior genealogists are responsible for assessing the work of the other genealogists on a biannual basis. So really the question is, which standards apply and do we need new standards? There's an emerging section under ANAB that falls under the scope now of SNP genotyping to include forensic investigative genetic genealogy. So that's a newly um, developed criteria for establishing standards, includes audits, and so this is moving in the right direction. And there's a few laboratories that are seeking or have obtained this kind of expanded accreditation. Okay, a little bit into now the technical requirements and some of the pitfalls, DNA extraction and library preparation. Okay, so what kinds of biological samples can be used? Well, it turns out that really anything that can be high enough quality and enough DNA will work. And as time has gone on and technological improvements have been developed, more and more DNA information can be obtained from smaller and smaller and highly degraded samples. So the the most difficult by far are aged bones, old bones, but they can be typed, and many unidentified human remains cases have been solved using bones. So even though it's difficult, a lot of progress has been made there. Hairs are tough evidence, although with improvements in library preparation techniques, hairs can now be fully genome sequenced. Something that was thought to be really kind of inconceivable just a few years ago. Hair, so tissue, muscle, buccal, and saliva, and now also touch DNA. So again, touch DNA are things like, whenever a person sweats onto something, like a ball cap or a steering wheel, clothing, that kind of thing. If a good extraction is performed and the DNA is high enough quality, it can be used for uploading and for SNP genotyping. So pretty much anything. There's a lot of improvements in DNA extraction technology using a lot of different types of chemical affinity between DNA and columns, magnetic bead technology, for example, very strong. The ancient DNA community has developed these many years ago, and now the forensic community has adopted them, and it results in very clean, highly typable DNA from improvements in extraction efficiency. Here's just one called silica-based DNA binding technology, very strong, very powerful, used in the ancient DNA world and now routinely used in forensic labs as well. Importantly, library preparation is important for taking a DNA extract and getting it ready for what's called whole genome sequencing, or WGS, whole genome sequencing. And that is it's not really an emerging technology, but it is emerging more and more as the go-to technology for these types of DNA extracts, whole genome sequencing. So what you really want to know is the three primary factors: how much DNA, how degraded is it, and is it a pure sample or is it mixed? So labs will ask all the questions about how it was extracted. Has it been subsampled? Has it been subjected to freeze thaw, etc.? Is it mixed? If it is mixed, then how mixed is it? Because some of the companies can handle DNA mixtures and separate out the contributors, and there's a lot of interesting recent work in separating DNA mixtures. So as time goes on and there are environmental insults, DNA, shown in its typical form on the left just falls apart. And so at some point it falls apart to the point where it's just not useful anymore. And in that case, sequencing technology is a better choice. And that's shown on the right. You can generate short little sequences from short little fragments, whereas the microarray shown on the left, that has a difficult time with degraded DNA. It can handle some degraded DNA, but not as well as sequencing. So for those really tough samples, like the bone sample, the degraded samples, those rely more and more on sequencing technology. So really, you want to know all the information that's relevant: the method, the kit, when it was extracted. Some of these go way, way back to the to the 1990s or sometimes before that. So typically you want to look at how it was how it was extracted and stored. But then you also want to use a modern technique to assess its quality. And so often retesting of an old extract is performed. So this just shows a DNA sequence. And then highlighted in the center there, you see that A that's a box. That's actually would be a SNP. And this entire sequence is 1,000 nucleotides in length. And there's approximately one SNP every 1,000 base pairs. So that's about the density of SNPs across the genome. And so what you're looking for is, that little bit of information that's different between individuals amongst most of the information, which is the same. It's conserved. There's no information there because everyone has the same type. But in those 1 in 1,000 instances where there is a SNP, then you can assay that and capture it, upload it to a database, and look for matches. And again, a lot of information, on and on and on, thousands of pages of just genotypes. And hopefully the call rates are high enough to where you can retrieve enough information that's usable. But again, degradation of the signal negatively impacts your ability to do that. Okay, a little bit on technologies, especially sequencing technologies. So in general, there's a cutoff between when microarrays are used. Anything above a nanogram, a high quality sample above a nanogram, generally up to about 25 nanograms, a microarray is used. Anything less than a nanogram of extracted DNA, DNA sequencing is used using a lot of different platforms. And there again is the same slide as before, talking about targeted kinship. The importance here is that the kinship testing, because it's high quality DNA, just goes on the microarray. So there's no need to sequence a kinship sample. And that's relatively fast, high QC, and relatively inexpensive. So for those types of reference samples and kinship samples, the array works just fine. And here's a picture of the array. There's different types. They're generally developed for the clinical medical testing world. And so a lot of these SNPs have some kind of disease associations or potential disease associations, and they have different densities. So forensic samples use the cytosNP 850K, and then for kinship samples, there are other density SNPs like the GSA array. You can load 24 samples per chip, and that's just to find the non-forensic, but just the kinship associations, the references, because you're looking for those relationships. Whereas in the top for the forensic samples, those will generate the actual unknowns from the forensic sample. So this is an overview of kind of what the technology looks like. So on the left, you have the bead chip and the array scanner. That's the microarray. And on the right, you have the DNA sequencer and its flow cell. So two fundamentally different technologies at play. So on the DNA sequencing side, it's very important to formulate or to create what's called a DNA library. The sequencer won't work on just naked DNA, on DNA that's extracted. It has to be prepared in such a way to create what's called a library. And there's a lot of different ways to create libraries. That's why I have highlighted here in different colors, red, yellow, etc. And this is where rapid advances, and just in the past few years, have improved the efficiency of creating a library from very small little fragments, and has allowed, for example, investigators to look into short little fragments in hair shaft, and with enough of those short little fragments, be able to stitch them all together and create a whole genome sequence from a person from a snippet of their hair. And you don't even need the root, it's the hair shaft. So that's an example of how a DNA library improvement has led to a huge forensic technological improvement. So DNA library prep, very important. That's where kind of the rubber meets the road. You've got this extract, it may be highly degraded. What can you do with it? Well, you make the best library that you can, and then take that library and sequence the library. There's also emerging technologies which sequence in different ways. This is an example of a nanopore DNA sequencing instrument. Here, the DNA passes through a pore, and as it passes through the pore, there's a difference in voltage that is a function of the sequence of the bases coming through. And so you can actually see the sequence of the DNA as it's passing through the pore. You can actually see it in real time. And it's just the signal is very small changes in current or voltage coming through that pore. So this is another potential application. It's not being used yet, but it could be very powerful addition to this application. Okay, so finally, funding. So there are a number of funding sites. The Bureau of Justice Assistance at DOJ is a good one. There's also the SACI program, also under Bureau of Justice Assistance. They have 4 strategic focus areas right off the top: reduction in violent crime. So this is a very good fit to the DOJ guidelines for investigative genetic genealogy, because that's exactly what they're trying to get through using this technology to solve old cases. So a lot of synergy, a lot of overlap between the mission of the Bureau of Justice Assistance and what genetic genealogy can do. Here's the website. If you want more information, there's a new solicitation that's out just now under JAG funding and also BJA funding. So check out the possibilities there for obtaining funding, state and locals. The SACI grants go to state and locals and a lot of local law enforcement agencies, and to address sexual assault backlogs. A lot of these can be addressed through genetic genealogy. You have a sexual assault case or a serial rapist. Profile has been obtained, uploaded to CODIS, no hit. So what do you do? What can you do? Well, one of the possibilities is to use genealogy to solve a number of your backlogged sexual assault kits. So specific funding just for SACI. Season of Justice is another very popular one. They run a podcast. EWU Media also can provide funding. They're big supporters of this technology. So now, real quickly, these are what kind of I see as some remaining issues, not all the remaining issues, but kind of what I see as what we need to think about. The first one there is to maintain strictly voluntary status of matching. This is all done voluntarily through database uploads and law enforcement buying into it, using it to help solve their cases. There are a number of guidelines to follow, but it's maintained so far. It's strictly voluntary status, and my personal opinion is it should stay that way. And most people that I've talked to agree with that. Another issue, though, is, these are private companies. GEDmatch is a private company and now owned by Verigen and Qiagen, and Family Tree DNA is a private company. They have their own terms of service. These are not government entities. They're not subject to any government regulations and laws. So again, it's voluntary. It's outside of the public sphere for the most part. Although now the FBI is significantly increasing their footprint in this area, and so there are publicly funded data law enforcement agencies that are solving these cases. But the database uploads to the GEDmatch and the Family Trees, those are still private companies. So even the FBI, a publicly funded crime lab, they have to abide by the private terms of service. And this is important because there have been allegations of some uploads into these databases outside of the terms of service, and that could be problematic in the long run. The other issue is maintaining the security of the raw data. So I showed you the match list, and the match list doesn't have any genetic information, but the genetic information does exist somewhere, in the laboratory, and maintaining the security of that data is important for long-term stability of this technology and the application of it. Okay, thank you for your attention. Don't know if we're going to take any questions, but I appreciate the time.
I don't see any questions. Mark, on behalf of the National Institute of Justice, National Center for Forensics, thank you very, very much.
You're welcome, Joe. My pleasure.